Thank you, Subit. Thank you, Dean Georgopoulos, for organizing this. Excited to be presenting in front of faculty and other stakeholders in the College of Engineering. My name is Swami Rajaraman. I'm an assistant professor in Material Science and Engineering and Nanoscience Technology Center. Our website is actually pretty up to date. There's a lot of information that I'm going to discuss very quickly here, given the 10-minute forum nature of it. So if there are questions, always happy to take it now or beyond. One thing I did not put on that I see that the other presenters had on is just a general background. I sort of took like an unusual route to academia. I got my PhD in electrical engineering from Georgia Tech, doing biomedical microsystems, but then I founded my own company and I worked there for some time. I was part of a team of co-founders actually, worked there for some time before I came back to academia to UCF in 2016. So just the introduction. So the research teams in my group are threefold. So first is the development of micro and nanofabrication technologies based both on things like 3D printing as well as more traditional semiconductor techniques for biomedical and agricultural interfaces. And I'll talk specifically about what those interfaces are. I'm just showing a couple of examples here. And along with that also goes studying the fundamentals of some of these techniques. So some of the fundamental techniques in things like 3D printing and other kinds of technologies that we use, like laser micromachining, are somewhat understudied, and we make some fundamental contributions there as well. I have a group of about 10 students typically, and about 20 students or so have gone through the program. A lot of them are EU students. I had a couple of students from Germany coming and doing their undergraduate thesis. Typically, I've had a couple of senior people in the group that sort of lead some of these different project teams. And of course, teaching was another thing we were asked to highlight. And I teach courses in fundamentals of material science to mechanical and aerospace engineers, as well as I've developed two graduate classes in laser materials processing, as well as an interdisciplinary class in biointerfaces, which covers a lot of the research that my group does. So what is the motivation for biomedical and agricultural microsystems and the themes of my research? So the motivations are in recapitulating a lot of the signatures that typically happen in the body to outside the body on a chip setting. So there is a lot of, I'm sure everybody's heard of things like organs on a chip and different models of organs on a chip. Specifically with respect to neurological models, there is still a lot of information that is relatively missing. And in order to recapitulate a lot of these signatures in a dish, we develop instrumentation, mostly sensors, micro and nano sensors, to interface with these variety of cell types in a dish. Of course, the current priorities, that national and international priorities, including potentially some COVID-19 in vitro models, the opioid epidemic, we've been funded by the NIH on that. So those are some of the motivations, in addition to testing new materials and systems with a variety of these cell lines, in being able to as closely as possible mimic in vivo conditions in an in vitro setting. As far as agriculture goes, I've had no experience prior to coming to UCF. And when I came to UCF, talking to a faculty member, Dr. Santra, in chemistry and nanoscience, and they were looking for delivery technologies to deliver this novel nanotherapeutic that he had developed in his lab into plants in a highly precise fashion. And that's where we adopted some of our Makerspace micro nanofabrication technologies to develop some of these interfaces. And once you develop a delivery technology, you can also use it for other kinds of applications, such as monitoring and other things. And also go beyond just bactericide and insecticide delivery, but also start looking at agrochemicals and the like. And going along with all of these, of course, is always looking at the fundamentals of some of these processes and incorporating all of these ideas into graduate classes, especially some ideas from the industry. I typically have, coming from an industrial background, I typically have some guest lectures from the industry during my graduate classes. So 4 quick topics that I would like to discuss in the next couple of minutes. As I said, I'm going to go really fast. So the first, and this is where most of my group, most of the energies in the group are spent, is in the development of 2D and 3D microelectrode for probing these electrically active cells and constructs. We have established relationships and collaborations with Tulane University, University of Wisconsin, and of course at UCF. and funding from NIH and some private companies as well. And the motivation, I've shown 2 examples here in the development of 3D printed 3D microelectrode arrays. That's the one that's shown on the right-hand side on the top, with enhanced functionality with things like nanoscaffolds and nanotherapeutics for control of bacterial colonies, for instance. That's the example that we showed there and published that last year in IEEE JMEMS. And the bottom example encompasses 2 different approaches that we have taken to not just make these nanomaterials for enhanced impedance and signal to noise performance, but also with Professor Hickman's group at UCF printing human motor neurons and human cardiomyocytes right atop the sensor, microelectrode sensors, and this was published in IEEE JMEMS as well. And also the other big theme of my group is to try and incorporate these makerspace fabrications and compare it to traditional fabrications. It's something that we do all the time. I'm not going to go over all the impacts and outcomes and achievements, but we published a few papers, and I'm a big believer in filing patents as well. So I published a US patent and PCT application on this. A real quick example on the agricultural application. So this is the delivery thing that we were talking about. There are all these efficient bactericides and insecticides that have to be delivered into plant tissue, but typical soil drench and foliar applications are just not sufficient for the amount of uptake that is required to treat some of these bacterial infections. So we came up with the idea of using microneedles, and I believe we are like the first ones, as far as we know, to have demonstrated microneedles for a plant-based applications. There's a couple more approaches that have come up to more recently, but we have additive manufacturing-based microneedles for treatment of leaves and stem and in younger plants, and then metal-based microneedles that we published in scientific reports on treatment of more grown plants. And we have even shown larger systems for like trees and such, where we've increased the uptake of these therapeutics by 6-7 orders of magnitude when compared to just pure diffusion. Once again, on this, we have published a journal article, and there is a patent application that's filed as well. This is more of an interesting thing that I just brought about because it was relatively new, and this is an area where I'm looking definitely for collaborators. So one of the things that I've noticed during my years of experience in microelectrodes and in vitro applications is there is no dynamic, that is mechanical movement as you're measuring electrical signatures. Of course, it's a difficult problem to address. So we took 3D printed architectures such as a microserpentine, optimized it analytically, then printed a bunch of these microserpentines, made them conductive, and created 3D sensors and micro LEDs on top of this. The idea being it can be used for anywhere from in vitro to wearable to potentially implantable applications. Again, this is something that we have published on, we have a patent on, and this was selected as a finalist for Microtask 2018 poster presentation, but it's something where I'm definitely looking for new opportunities to collaborate and new opportunities for applications in this space. And then lastly, since I'm running out of time, I'm gonna showcase another thing that we worked on is, typically microelectrodes have a conductive layer and an insulating layer on top of it, whereas interdigitated electrodes are these simple finger type electrodes or circle and line configurations that just have a conductive layer, and they're typically used for studying morphological changes in cells and studying drug responses and the like. So what we did, and working with Professor Thomas at UCF, is taking a nanostructured substrate and defined interdigitated electrodes on top and compared it to more standard approaches. And we were able to show multiple orders of magnitude better performance compared to standard approaches. And this also was accompanied by work in laser micromachining and being able to show some of these ablation characteristics on different materials. This again is the subject of a couple of journal papers and one filed patent. The near-term and long-term objectives, including consolidating the current funding teams, the current research team procuring funding. I'm working on submitting proposals both to USDA and NIH. I'm also working in ECE with the MIST Center and try to bring more companies into the MIST Center for company-funded projects. Graduating the students that I have, including a couple of PhD students, and establishing new collaborations with other organ-on-a-chip people in Texas Tech and Johns Hopkins. And the long-term goals are, of course, to develop more lab-based classes and also to do more system-level stuff. We can do a lot of sensors and a little bit of biology, but we would definitely need help in computation and other kinds of advances on top of these systems. And with that, I would like to close acknowledging funders, all the students that do all the hard work, the collaborators, and then open it up for questions. Thank you.